Now comes the long patch repair. So the alternative pathway to repair the damage in mammalian cells is termed as long patch repair. In this pathway, 2 to 8 nucleotides are replaced in contrast to one nucleotide which was replaced in case of short patch repair. In this case, DNA polymerase delta or epsilon catalyzes chain extension with the assistance of the RFC, that is replication factor C clamp loader, and the PCNA sliding clamp. So in this long patch repair, DNA polymerase delta or epsilon is used, which causes the extension of the chain instead of pol B or any other polymerase. As the polymerase adds nucleotides to the 3' hydroxyl end on one side of the gap. So this is a strand. So on this side is a 3' hydroxyl group. So the polymerase adds nucleotides to this side. It displaces the 5' deoxyribose phosphate and on the other side of the gap. So as the new nucleotides are introduced on this side, they are added on this side. This on this side 5' deoxyribose phosphate is slightly displaced. This strand is gradually extended and this strand is gradually displaced. So in this way, in this fashion, it goes on. Then the flap endonucleases cleaves the displaced strand and the DNA ligase finally seals the remaining nick. So this strand, this is newly synthesized and this one is displaced. So this portion may contain 4, 5, 6, 8 nucleotides. So the flap endonuclease then cuts this displaced portion, and finally the DNA ligase uh, seals the nicks. The regulatory mechanism that selects long or short patch repair is not well understood. If there is some damaged DNA, the regulatory mechanism will select either short patch repair or long patch repair. What determines this? This is not clear. So here you can see this is a long patch repair. Again, this is the damaged DNA, which this base is excised by DNA glycosylase, and then a P endonuclease introduce a nick here, and then pol delta or epsilon adds new bases, new nucleotides here. These new nucleotides have been added to 3' hydroxyl end, and this strand, this one, this is gradually displaced. So after that, flap endonuclease cuts this from here. Flap endonucleases cuts this displaced strand, and now this happens, and finally this gap is filled by DNA ligase. So in this way, a damaged base is excised and finally this DNA is repaired. So this is the summary of long patch repair. When eukaryotic base excision repair begins with a DNA glycosylase lyase, the pathway is shown below. So this is the pathway in the eukaryotes where the repair has been started by DNA glycosylase lyase enzyme. So this here you can see this is the damaged base, and this the base is excised by AP glycosylase lyase, and then because it has dual function, a nick has also been introduced here. Then AP endonucleases cuts this, and then pol B adds this deoxyribonucleotide to the 3' hydroxyl end, and finally DNA ligase seals the nick. So in this way the damaged base is repaired. So three distinct glycosylase lyase enzymes have been identified in E. coli. Three such enzymes have been identified in Saccharomyces cerevisiae, while six such enzymes have been identified in human cells. Virtually all the oxidized bases are removed by bifunctional DNA glycosylases in mammals. DNA glycosylase lyase excises the damaged base and cleaves the DNA strand 3' of the AP site. So on the 3' of the AP site, the strand is cleaved by this DNA glycosylase lyase. The resulting sugar residue at the 3' end is removed by the AP endonuclease catalyzed cleavage. So AP endonuclease cleaves the sugar remained at the 3' end. Then DNA polymerase beta adds a nucleotide and DNA ligase finally seals the remaining nick to complete the short patch repair. So here you can again see the same pathway. This is the damaged base, and after a number of steps it is completely repaired with a new nucleotide here.